Hello! Today we're going to talk about all the sensory organs involved in neurophysiology. So, as you're probably well aware from childhood, there are several senses that we deal with: hearing, sight, tasting, smell, and touch. And today we're going to look at each of the organs that are involved with those senses, how they work, and how they convert the information, whether it be sound waves or light waves or pressure or chemical senses, how all of those information is converted by your sensory organs and then sent to your central nervous system for processing and understanding of what it is that you're sensing. So first of all, we're going to start off with the skin. The skin is your largest organ in your body, and it contains highly specialized neurons that are specially designed to measure certain things. For example, your Pacinian corpuscle, that looks like this, so it's a dendrite ending with myelinated sheaths around it at the end, senses vibration. So if my hand is, for example, on a solid rock versus it being on the hood of a tractor, right? The amount of frequency between those two are widely different. The rock, if it's solid, you're probably not going to be feeling anything. If you're, driving an old truck and you put your hand on it and it's vibrating, you can easily feel that vibration. But the reason why you're sensing that vibration is because your Pacinian corpuscles are changing shape at the end of that dendrite, and it's telling you that there is a vibration. Then you have the Rossini corpuscles, and the way that they're aligned is along the length of the skin, and so they respond to skin stretch. So if I'm bending my wrists, for example, my skin is stretching on the one side of my joint, and my Rossini corpuscle is firing, saying that that piece of skin is being stretched or not. Merkel receptors senses steady pressure. So if you have your fingers, say, on the table and you're just lightly resting them versus pressing down hard, it will tell you how much pressure you're putting. So if you can recall back to the first time you ever used a iPhone or iPad, for example, and the amount of touch that you have to give it, right, is so much different than if you're actually using a keyboard interface. And so the amount of pressure that your body senses and know that it's required has been incorporated from your Merkel receptors. The Meissner corpuscles, that's these fellows over here, they respond to stroking movement. So if I'm stroking my hand over a piece of velvet versus the ground. And then free nerve endings, usually the free nerve endings are the ones that senses pain. If you have touching a hot stimuli or something sharp or something like that, the nociceptors at the end is going to be stimulated due to the pain that it's experiencing. And so all of these specialized sensory organs at the very end, they all, via sensory nerves, send that information from the sensory organ to the central nervous system via the spinal cord. Here's a summary of pretty much what I just explained each of them are and where they can be found and what they respond to. Now, temperature receptors are very interesting, and they're also part of the free nerve endings of your body, and they can sense cold touch versus warm touch. So, for example, if I put my hand in a freezer or if I put it onto a heating plate or the edge of a really hot styrofoam cup. Okay, my free nerve endings will be sensing that. And what they do is they correlate that to your core body temperature to say, okay, well, how's it doing? And pain receptors are going to activate above 45 degrees Celsius. Now, the neural pathways for these are, whether it's fine touch, proprioception, or vibration, it's going to come into your dorsal root ganglion, and then straight into the white matter, and then it's going to go up through an ascending pathway. In your medulla, it's going to synapse onto an interneuron that is then going to take that information up to the thalamus and then to the motor cortex. Okay, so pain, temperature, coarse touch, all of those, so the nociception ones, okay, they're going to come in through the dorsal root ganglion. They're going to have an interneuron, though, that they're going to synapse onto on the gray matter within the spinal cord. Then they're going to also go up to the medulla, but they don't split there. They just go straight up to the thalamus. And in the thalamus, the sensory pathways are going to synapse, and then they're going to go off to the somatic sensory motor cortex, where it is going to interpret the information that it has received. The somatosensory cortex, okay, each body part has its own unique location. So the face is over here, the hand is over here, the back and the foot. And as you can see, the biggest organ for the somatosensory really is the arm and the face. And if you think about it, right, with your fingers, you need a lot of dexterity and a lot of control over what it is that you're sensing and doing and touching and feeling. And so that's why that takes up the most part of the somatosensory cortex, versus your elbow or your back that not that much. So this little diagram here shows kind of along the lobes how it is spaced. Next, let's talk about the olfactory system, or the nose. The olfactory pathways is the fancy word for the neurons that sit right up top in your nasal passage. So this is the hole at the front of your nose. The air comes in through here and goes back through the back. Any particles or anything that is chemically in the air or so forth, it is going to bind to these olfactory neurons here. And they then are going to all sum up here in what is known as the olfactory bulb. And the olfactory bulb is going to send information via the olfactory tract to the olfactory cortex. And the olfactory cortex will then let the cerebral cortex and the limbic system know what it is that you're smelling. So let's take a closer look at how those nerves work. Down here I have my nasal passage, and I have a mucus layer over my free ending neurons there. The reason I have the mucus layer is both to trap any chemicals or any stimuli that are coming in, but it's also to keep those neurons moisturized so that they don't dry out. And then you have the cilia or the dendrites, right? And then you have the supporting cell layer here, where it contains the olfactory sensory neurons, and information so travels from here up into the lamina propria, through the bone that is up in your part of your skull, and then it goes up to this becomes the olfactory nerve. So the odorant is going to bind to some of the receptors at the bottom here of your free ending. That's going to cause an action potential chain that's going to send up, and then your brain is going to recognize that this smell is X. So here's another view of it. You have your olfactory sensory neurons here, you have the bone here, and then you have your big olfactory bulb here, and they all synapse onto the secondary sensory neurons. So taste or gustation is closely linked to olfaction. In fact, if you have a stuffy nose, you've probably experienced this before, but if you have a stuffy nose and you try to eat something, you might have a difficult time distinguishing between two different tastes. The reason being is that smell is such an important part of eating, and the smell tells you more distinctly what it is that you're eating, where the tongue is really used to break it up and send collective information. So taste is going to be a combination of only 5 different basic sensations: sweet, sour, salty, bitter, umami. Okay, and each taste cell is sensitive to only one of those 5. So here you can see here's a cell that's sensitive to sweet, one to umami, one to bitter, one to sour, and the last one we don't really know, but we think that that is the ones that are activated due to salt. So let's look how taste transduction work. You have ligands that are going to activate the taste cell. So if you have sour, for example, you have an H plus molecule, hydrogen molecule. It binds to the cell, the receiver cell at that. Then various intracellular pathways are going to be activated. Calcium is going to flood into the cells. Okay. And then this is going to trigger exocytosis, on this case for sour, or is going to cause ATP formation to occur in the case if you're stimulating sweet, umami, or bitter. And this is how you know whether something is sour or if this is something is sweet or umami or bitter. It's whether it's the ATP that's being formed or whether it's the neurotransmitter that are being released. And so this is how your brain distinguishes between sour and sweet. And then lastly, the primary sensory neurons are going to fire and action potentials are going to be sent to the brain. The ear is a little bit more complex than the tongue and smell, and it's because it has so many more different parts. The ear is really divided into 3 sections: the external ear, the middle ear, and the inner ear. The external ear consists of the pinna, which is the shape of your outer ear, which is the part that you can see, and it is shaped in such a way so that it optimizes sound waves traveling into your ear canal and that are going to vibrate your tympanic membrane. This tympanic membrane, when it vibrates due to the sound waves, is going to be changed into a mechanical energy. So we're going from sound energy to mechanical energy by then vibrating 3 little bones in your middle ear called the malleus, the incus, and the stapes. They are in turn going to vibrate against another tympanic brain called the oval window of your cochlea, and we'll look at the cochlea in a little bit. And the cochlea then has special neurons attached to it that senses the sound coming through it, and it then sends that information off to the brain. Now, hearing is our perception of energy waves as they come in, right? And energy waves have wavelengths, so they have peaks and they have troughs. And the frequency is going to be translated into pitch, and loudness is the amplitude of it. Now, let's walk you through this process. You have sound waves that strike the tympanic membrane, and they become vibrations. The sound wave energy is transferred to 3 bones in the middle ear, the malleus, the incus, and the stapes. Then the stapes is attached to the membrane of the oval window, okay? And the vibrations of the oval window is going to cause the fluid which is inside your cochlea to now cause little waves as it moves. The fluid waves are going to push on the flexible membranes on the cochlear duct, okay? And hair cells that are in this duct is going to bend, and that bending, that mechanical bending, is then going to open ion channels that is going to create the electrical signal and alter the neurotransmitter release. The neurotransmitter release onto the sensory neurons is going to create the action potentials that are then going to travel down your cochlear nerve to your brain. And lastly, the energy waves that are in excess are going to be transferred to your round window and back out to your middle ear. Now the cochlea, if I took the cochlea that is like this and I completely unfolded it, I would get this shape. And so let's talk about anatomy real quick. I have the oval window here. This is where the wave starts. I have the vestibular duct, which is this top part, the cochlear duct, which is this middle part, and the tympanic duct, which is the bottom part. And I have a basilar membrane here that separates the vestibular duct from the tympanic duct, and then down here is the round window where the sound then dissipates back into the middle ear. Now, in your middle ear you have these highly specialized cells that sit within your cochlear duct, but have little protrusions out into your vestibular and tympanic duct. So let's take a look at how these cells work in order to change that mechanical change or vibrations in the fluid into chemical changes that then cause an electrical impulse on your neurons. Now, at rest, so this first one here is rest, about 10% of the ion channels down here is open, and so neurotransmitter is steadily being released. And so you see here on my action potential that I'm measuring here, I have a steady rhythm, right? And that's due to the constant release of neurotransmitter. Now when excitation occurs, so that means the wave is traveling from left to right, that's going to push these little hairs here to the right. When that occurs, more channels are going to open, so more neurotransmitter is going to release, so there's going to be an increased frequency of the cells firing. For inhibition, this is when the fluid is moving in the opposite direction. The channels are going to close, and when they close, there's no signal that's being transduced. And then we return back to normal at rest, where we have a steady pace. And so this is how your body knows this is normal, this is excitation, and this is inhibition, based off of the frequency of the action potentials that are traveling down your primary sensory neuron. Now the basilar membrane also has variable sensitivity to the sound frequency along its length. So when the sound first comes in to the cochlea, that is where all your high frequency sounds are going to be heard. And then as it travels down, it travels from a high frequency to a low frequency or a low pitch. Now once the information signaling leaves the cochlea, it travels to the medulla, where there's the cochlear nuclei, and then it goes up the ascension pathway to the midbrain, and then to the thalamus, to the right and to the left. And you can see here, there's a crossing that occurs as well. So, if I hear something coming from my right cochlea, I'm going to receive that information in my right auditory cortex, but also my left auditory cortex. And the same if it comes from the left cochlea, it's going to be sensed in both ways. The reason for this is because this crossing helps your brain identify which direction the sound is coming from right or left or backwards and forwards. That's why if you were to listen to, say, a police siren as it comes by, or an ambulance siren as it comes by, it changes frequency and pitch to you seemingly as it moves from, say, behind you to the front of you. And the reason of that is because of location relative to you. The sound itself doesn't change, the wavelength itself doesn't change. It just changes in relative position to you, and that's what your body is sensing. Now, besides hearing, the other important thing that your ear is responsible for is maintaining your equilibrium. And meaning that you're able to stand upright, or that you're able to sit upright without having to think about it. And your ear does this with very specialized crystals or crista that are located within the semicircle canals of your ear. And there are 3 loops. There's the superior loop, the posterior loop, and the horizontal loop, with these little crista that are located in it. And here's an image of it, okay? So the crystals are going to be lying on top of a gelatinous otolith membrane. And then when your head bends to the right or to the left, these crystals are actually moving. So it's a mechanical change, right? And these are changing due to gravity pulling on them. And that changing is going to change the distribution of them all across your gelatinous otolith membrane. And that is going to bend these little hair cells that are located there. And that bending, once again, is what's going to cause an action potential to occur within the neuron. Equilibrium pathway. So once all of that summation has occurred, the information is going to travel down the vestibular branch of the vestibular cochlear nerve. It's going to go to the cerebellum, and then from there it's also going to go to the vestibular nuclei of the medulla, up to the reticular formation, thalamus, and the cerebral cortex. Remember, this has to do with motion, not with hearing, so it doesn't go to the auditory cortex. It goes to the cerebellum, where all motion information is synthesized. And then it goes up to the cerebral cortex, because now your brain has to know, okay, well, am I bending too far to the right? I need to adjust. Or this is also why, if you've been on a boat and you have your sea legs, right? That's what they say, sea legs. It's just because your motor cortex has learned to compensate for those changes of the waves of the boat. So that's why when you get back on land, you have to readjust. It's because the brain now has to interpret the different shifting of these crystals and how they work. Alright, so that sums up the ear. Next, we're going to look at the eye. Now, the eye, where the ear is complex, the eye is even more complex, and the reason for that being there are so many minute little muscles all around the eye to control the eye's movement, not to mention all the specialized cells to help distinguish between colors, between distance, and all those sorts of things. So, light and dark. So we're going to be looking at that next. First, let's look at the external anatomy of the eye. You have the upper eyelid, the lower eyelid, the iris, which is the color part, the pupil, which is the black little part in the middle, the sclera, which is the white area surrounding your iris. You have the lacrimal glands. They're going to be secreting tears. And you have muscles attached to the external surface of eye for control movement at the top and the bottom, and actually beside the sides, but you can't see this on this picture. Okay, and all of this, the eye is located within the bony cavity of the skull, which is meant to protect the eye. Looking at the eye from the side, you can see all the muscles I was talking about from the side, from the top, and so on. But let's take a closer look. So all I've done is I've cut the eye through in the middle, and that's what this picture is showing. Where to start? Let's start from the outside in. So at the very, very edge of the eye is what is known as the cornea. And it is a very thin film that covers the eye. This here is the pupil that will change the amount of light entering the eye. So the smaller the pupil is, the less light goes in. The larger the pupil is, the more light goes in. That's why, if you were to look, well, that's why when it's dark and outside, you have larger pupils. The iris, this is the color part, okay, so is right next to the pupil. And then behind that, you have the lens. Now, what the lens is going to do, and we'll talk about this, is it's going to bend the light so that the light can focus on the back here where all your highly specialized neurons are. That's going to interpret the information from the light. Then inside the eye, you have the vitreous chamber that contains liquid. And then you have the retina, which is the layer that contains the photoreceptors. And the photoreceptors are those specialized cells that detect the light and detect color. Right here in the back, you have what is known as the optic disc, or your blind spot. So this is the region where veins and arteries come in and leave your body, and leaves your eye. The reason for that is because they come in, there's no photoreceptors in that area, and so your photoreceptors, since they're not there, that part of the eye can't sense what is being looked at. That's why it's called the blind spot. Then you have the optic nerve that takes all of that information up to the brain. Here's a specialized ophthalmoscope vision look at the eye. You can see here's the optic disc. Here's your fovea, or your focus area. So on this view, all I'm showing here is you have the optic tract runs. So the optic tract runs from both eyes, and they meet at a central location, both both optic nerves. They meet at the optic chiasm, and then they split off again into the optic tract. And the optic tract is what runs into your brain. So here's the pathway. We have the eye, the signal goes to the optic nerve, then the optic chiasm, then the optic tract, then to the thalamus. From the thalamus, it goes to the visual cortex. Here's a different view of how that looks, but you can see here. What is interesting to note on this slide, once again, is just that there's a crossover of neurons and signals that occur here. I think I have. Oh no, I don't have a better picture. So anything here, right, on this side, that lands here on the right side of the eye, is going to go to the right side of the brain. If anything is towards the right and it lands on the left side of the brain, it's going to go to the left brain. Same here. If it ends on the left side of the eye, it'll go to the left brain. If it ends on the right side of the brain, it'll go to the right side of the brain. And so that is how your eye distinguishes where in space something is, by having two reference points, because you have two eyes versus one eye. This is also how depth perception works, is because your eye is able to distinguish where in space it is. For people that are blind in one eye, they lack that depth perception because they don't have that second reference point to tell them where the object is relative to them in space. Now, the function of the pupil is that light enters through it, and the size of the pupil is going to modulate the amount of light that reaches the photoreceptors. It also has pupillary reflexes. So, for example, if you're lying in the dark and somebody flips on the light, right, instantly your pupils are going to contract without you even having to think about it. And the reason for that is because it's a reflex. The lens, that, as I said, is responsible for directing the signal, and let's take a look at that. So if I have a concave lens and I have parallel light rays coming in, what is a concave lens going to do? It is going to split the rays further apart. That doesn't really do me any good in the eye. So for the eye, what we want is a convex lens. That is going to streamline everything to a single point, and that point is called the focal point. The focal length is simply the distance between your lens and your focal point. So if we impose that upon our eye down here, you can see that the light coming in from the outside is going to hit that lens, and so the lens is going to want to have a convex shape so that it's able to focus the light rays at our focal point. And the distance between our lens and our focal point is our focal length. Now, for close objects, okay, the light rays are no longer going to be parallel as they come in, and so the lens and its focal length, right, have not changed, but the object is seen out of focus because the light beam is not focused on the retina. So you can see here for closer objects, the focal point ends up being the back, even though the focal length, that's why it's kind of out of focus. And so to keep an object in focus, the lens must change shape. It must become more rounded. And so that is a term called accommodation, the process by which the eye adjusts the lens to shape to keep the object in focus on your focal point. And near point presbyopia is the loss of this accommodation, and so people with presbyopia have a difficulty focusing on objects and everything looks blurry to them. So the lens itself is attached to a muscle by inelastic ligaments called zonules, okay? And the muscle is called the ciliary muscle. And so when it contracts, if it's in a relaxed state like it is right here, your lens is going to be more flattened and it's going to function well. And then when the muscle contracts, these cilia are going to loosen up, and so that's going to allow the lens to become more rounded, therefore changing the focal point. Myopia or nearsightedness occurs when the focal point falls in front of the retina, and so that's why you then have to wear glasses that are concave, that is first going to split the rays apart so that your eye lens can then convex them back to the right position. And that's why you wear contacts or why you wear glasses, is to have that effect. Hyperopia or farsightedness occurs when the focal point falls behind the eye, and once again that can also be changed by having a more convex lens in front of the eye that's going to streamline that change in rays direction. Now let's talk a little bit about color. So the electromagnetic spectrum, you can see, is really, really large, from gamma rays all the way down to radio waves. But for humans, the visible light that we're able to see is in a very small range right here. And we can see colors from purple to red. And that the color change is due to a change in the wavelength, and that's the amount of energy that the wavelength has as it's hitting the eye. Now, phototransduction is the process by which the specialized cells in the eye measure the color and darken light, and then sends that signal to the brain. So it's going to convert light energy into electrical signals, and it's going to use photoreceptors. And there are two kinds of photoreceptors, rods and cones. Alright, we've talked about this, so I'm going to skip that. So let's take a look at the back of the retina here. Okay, so this is, this right here is where your optic nerve is with your optic disc, so your blind spot. And then next to it, the sclera is the back of the eye that completely surrounds the eye. It's like the part that you can see if you took out the eye. And then you have the choroid layer contains all the blood vessels that all of these receptors are going to need, and then you have the neural cells of the retina, which we're going to take a closer look at that. Starting inwards again, I have my pigment of epithelium cells, then I have cone cells, and cone cells are used to detect color, and you have rod cells which detect dark and light. Then you have a bipolar cell, which is kind of like an intermediary cell, and the cell where the light hits first, you have the ganglion cells. Right, so light hits from this side, it's going to hit your ganglion cells, then the ganglion cells are going to synapse onto the bipolar cells, and then the bipolar cells are going to synapse onto your cones and onto your rods, that is then going to synapse onto your optic nerve. this right here, this is the blind spot, and so light comes, sorry, not the blind spot. This is your fovea, or your focal point. So the light hits here and right there, and then it kind of disperses and you can see everything. So, the way that a projected image works, say I have a right here, the ray is going to come in, and then it's going to shoot up and you're going to see it on the left side of the eye. If it's coming in from the left, you're going to see it on the right side of the eye, and then you have this whole, spectrum. Alright, so let's talk about how rods converge information. So, the light hits, it's going to hit this pigment of epithelium. That's going to change the rod. The rod's going to affect the bipolar cell. The bipolar cell is going to synapse onto the ganglion cell, and the ganglion cell is going to become the optic nerve. Sorry, I messed that up earlier. Because, see, here's the tricky part. When the light comes in, the first thing it hits is the back end, and then it gets transduced forward. Alright, so, photoreceptors. So rods are functioning dark and light. Cones are responsible for color. And I need to talk about this. Okay, here we go. This is what I'm looking for. Now, we've got rods for black and white and cones for color. So here, for the rods, you have dark pigment epithelium. It's going to absorb the extra light and prevents light from reflecting back and distortion vision. You have all discs at the tip of the phagocytes by pigment epithelial that's going to sense that change. These discs look like this, and they have highly specialized rhodopsin molecules that's able to detect the amount of light being reflected back. Then you have mitochondria here that does the works in rods, and ultimately it's going to synapse onto your bipolar cell. Alright, this is just to talk about some people have color blindness, and they might be colorblind for red and green, but they can see everything for blue. And so if you can see blue, you can see violet through greenish, yellowish. But then if you're colorblind to red, you can only see up to the spectrums. So this is the reason of how colorblindness works. Alright, let's focus now on the chemistry. Now, in darkness, the rhodopsin that's in the cell is going to be inactive. Alright? And these little discs. And the cGMP levels are going to be high, and the CNG and potassium channels are going to be open. So potassium is going to diffuse out, and calcium and sodium is going to diffuse in. And so you're going to have a tonic release of neurotransmitter. And so this is if it's inactive, right? Okay, and then when the light hits this back end, it's going to bleach the rhodopsin. And so opsin is going to be decreased is going to decrease the cGMP. It's going to close your calcium and sodium channel. Thus, this means that you're only going to have potassium going out, right? And so your membrane is going to hyperpolarize. And thus, your neurotransmitter release is going to decrease in proportion to the amount of light. And then there's a recovery phase at the end for the retinal recombines with opsin and so forth. And so that's why this is another reason why it takes you a little bit of time to adjust, whether you're in the light or in the dark, is because it takes time for the recovery phase to occur depending on which direction you're moving in. Okay, so this is in darkness. You have a steady release of neurotransmitter, and then if you have light, you have reduction in neurotransmitter, and it's proportional according to the amount of light. Now, for how the pathway works is you have the rod, and then that's going to synapse onto your bipolar cells. Your bipolar cells are going to summative up to your ganglion cells that are going to go to your optic nerve. and we've already talked about this distribution, so I'm not going to mention that again. Okay. Now I just have a quick assignment for you guys to think about. What are the design criteria for a functioning robotic arm? There have been a lot of advances recently in creating, orthotic arms. It started with sci-fi, with Star Wars and Terminator and so forth. But in reality today, there are highly functioning robotic arms. So, I would encourage you to take about 10-15 minutes to think about just kind of like how you would connect up the actual hardwiring of a robotic limb to your nervous system. Thanks for listening. Bye.